visiting us for this couple of weeks with a Fulbright Fellowship program. So we are really happy to have you here. Professor Eric Ziblon is a professor at the Department of Ecology, Evolution and Behavior at the University of Minnesota. And he's also the director of the Cedar Creek Ecosystem Science Reserve, one of the first long-term ecological research sites in the US. And indeed, he will give a talk on next Tuesday at noon related with long-term ecological research and an overview of the US LTER, this is the name, the Long-Term Ecological Research Network in the US, and focus on Cedar Creek science. The areas of interest of Eric are community ecology of disease, diversity and composition of within host microbial community. He also did a lot of research on biological invasions. But today, Eric is going to share the research done in the NutNet, or the Nutrient Network, which is one of the coordinate research network. And that's how we met Eric, because we joined this coordinate research network about 12 years ago, when we had an experimental site in our Pampas grasslands in Las Chilcas. And that's how we met Eric and Elizabeth Borer. They are both one of the founders of this wonderful research network. So we are happy that he's going to share some of the science, amazing science that have been done in this network. And that's how we met also Anita and Martin, that are also visiting us this week. And we are happy to have you too. You heard Anita's seminars, I remember last year. Yeah.
Okay.
All yours.
All right.
Thank you, Eric.
Thank you, Laura. It's great to be here. And let me just start my clock here so I remember the timing. Okay. It's really wonderful to be here. First, thank you for listening in English, but with my bad Spanish, it would be a very short talk if I did it in Spanish. But there you go. Yeah, it's great to be here. Great to see old collaborators. It's been really fun to be working in person with Sophia and Pedro and Laura. Also fun to be meeting with people already. We had a great class the last 2 days. Good to see some of the people who were in the class. It was really fun to meet some of the graduate students and postdocs. Like I said, Laura said I'm around next week, so talk to Laura. I think she has my schedule. So I'm going to have sort of two overlapping themes in my talk today. So one is sort of a bigger, more general question of how do we study global change as researchers? How do we approach this problem? What are some of the ways we've done this in past? And then some of the trade-offs and different approaches. And the second is to take this case study of this research network that some of us started in 2006, and that Laura and Pedro and Sophia and Anita and Martin and collaborators all around the world have been coming together and working on this, and it's become a real model for a lot of other networks. So there's kind of, some sampling of some of the things we found, but also some kind of thought kind of parts of how do we do these kinds of experiments. So the main I don't think this one's working. There we go. All right. So kind of the context here, humans are having big impacts on the environment. This is a paper. There's been a whole string of these kind of papers, but this is looking at this idea of global planetary boundaries. But the main thing is humans are affecting the globe in many different ways. Big ones, we're increasing the fluxes of biologically limiting elements like nitrogen and phosphorus into these systems. So there's been about an 8-fold increase in phosphorus flowing into the oceans, doubling of nitrogen cycling. We're also causing climate change, changes in genetic diversity, species losses, increased invasion, and all of these sort of changes that are happening across the globe all also interact with each other. And understanding how these work can be really challenging because humans are having these impacts on these global ecosystem processes like biogeochemical cycles or climate, but they act through sort of organismal level processes. So changing plant physiology, changing interactions between plants and pathogens, between herbivores and plants, and then we have this feedback. So we have this cross-scale sort of process, and this can make it really challenging. Part of the challenge arises because these impacts aren't homogeneous across the globe. So this is a map showing the nitrogen deposition. So darker is higher nitrogen deposition, and I said there's a doubling of nitrogen deposition across the globe, but you can also see it's highly variable. So there's going to be a lot of context dependent. So you have to account for all these different sites, get different levels of impacts. The same is true for invasive species, for species extinctions, anything humans are doing, it's really variable across the globe. Climate change. And as individual scientists, we can only sort of work at a certain kind of scale, right? So we work at our sites, like you go out to Las Chilcas, you can do some work there. That's what we can do, right? We're limited, but we want to make inference at these bigger scales. And obviously, research budgets are finite, so we can't just do experiments everywhere. So this creates a lot of different challenges. Where we have people working at local scales. Ms. Lauren Hallett, who is at one of our study sites. She was here last year, right? We can do these field experiments, but then we're trying to create these inference across this highly variable and large globe, which creates this kind of conundrum for us of can we produce a sort of predictive and mechanistic understanding of how humans are impacting the globe. So if we're going to do that, and we have different approaches we might have, and there's some kind of properties that will help us make that kind of inference. So if we're combining data sets that use the same consistent methodology that makes things easier, if people are measuring things in different ways, in different places, that can be challenging. There's issues of causal inference, so working with experimental data, for example, you know what's causing the change. It's easier to make direct inference on causation. There's big observational data sets, and there's approaches to try to get causal relationships out of it, but it's always a challenge to go from correlation to causation. We also have, the real world is very complicated, so we can work in a greenhouse, work in the laboratory, can work on your computer and do simulations, and those are all useful, but those are really simple systems. We need to find ways to also scale back to sort of all these complicated things that are happening at the same time. Like I said before, impacts are variable across the globe, so there's really variable climate, there's different nitrogen deposition, different levels of invasion, so we need to know something about scaling this up across the globe. But also every system's a little bit different, so you need to think about doing studies that are tuned to your individual system. So not all kinds of experiments or treatments are going to work everywhere. And this hasn't been lost on ecologists, and we have sort of a toolbox of different approaches that we might use. Some we've used for a long time, so kind of the standard that many people have probably done are single site experiments. So you go out to your field site, you do some manipulations, and we've been doing this for 150 years or more. So that's nice because the methodology is consistent. Hopefully you're measuring things in your different plots in the same way. It's an experiment, so you can make causal inference about it. You're dealing with a field study, so you're accounting for all these other processes. You can tune it to your system, but then kind of like I started, how do you scale up from that? It's easier to do observational networks across bigger gradients because you're not trying to do experimental manipulations. So then you can get a bigger sort of network, but then you're stuck with sort of observational data. So all of these different approaches have different trade-offs, different challenges. There's also modeling kind of approaches we can use, right? So we can simplify the system, for example, mechanistic models, either calculus-based or more simulation models like Earth system models or ecosystem models. So that can help us out. And then the causal inference part, right? Because at least if you understand the model, you know what's causing what. But now we have all models are highly simplified. So now we're working in a much more simple system. Same with, you can build predictive statistical models, but then you're sort of sometimes sacrificing causal inference. Starting in, maybe in 2000, late 1990s, there was a real push in increasing synthesis in ecology. So using tools like meta-analysis, where you can take all these single-site experiments that people have been doing for many decades, pull them together, even though they have different measurements, slightly different treatments, there's some statistical techniques, you can pull them together and gain some larger insight. But there are problems because the methodology isn't always the same. So different size plots, different levels of treatments, which can lead to some challenges in terms of making inference. What I want to talk about today is sort of an approach that arose. So some of the people who founded this network were involved in a lot of these meta-analyses, and this is an approach that basically arose out of working on meta-analyses that could kind of complement this. And one of the points I want to take home is not like this is better. We need all these tools, right? Because they all have different trade-offs. And so if you take different approaches and they're all pointing in the same direction, then you can feel more confident, more robust kind of results. So if your observational studies and your experiments are lining up with your models, then we need multiple approaches here. But every one of these has different trade-offs. And so we wanted to look into an approach where we'd have some of the benefits of experiments, but also be able to make some bigger scale inference. And these are distributed experiments. So this is the nutrient network we started 2006, 2007, and there's been a lot of new ones that have started. So it really seemed like it was answering a need in our field. So there's studies like DroughtNet that Laura's been involved in. There's a warming experiment in the tundra using these open-top warming chambers. There's been marine ones looking at herbivore-nutrient interactions in seagrass beds. A lot of these things have been happening now, and a lot of new insights have come out of that. So I'll kind of switch and sort of move into this case study now for this one network that we've been coordinating since, for almost 20 years now, so nutrient network. And so the goal of this, the scientific goal, was to understand the global scale changes in both nutrient inputs and changes in the food web. So this is kind of one of these top-down, bottom-up kind of settings. So we have ecosystem properties like productivity, carbon sequestration, diversity, species community composition, maybe applied things like species extinctions or species invasions. These are affected by limiting nutrient supply. So in the case of plants, elemental nutrients like nitrogen, phosphorus, potassium, but also affected by trophic interactions, so by consumption. And in our case, looking at application of different limiting nutrients, nitrogen, phosphorus, and excluding grazers. So the motivation, like I said, came out of working on a bunch of these meta-analyses and to try to find new approaches that could complement sort of the suite of existing tools that we have to study human impacts at a global scale. And so we started working on developing a global network of collaborators that could implement a low-cost experiment. So we were all very, early postdocs, grad assistant professors, and also wanted this to be very approachable for people without having to wait around for a big grant, and something that would follow a standard methodology. So we decided to work in grasslands because they're the most interesting ecosystem. Well, not, of course, exactly, right? Why grasslands? There's lots of different ecosystems one might work on. Grasslands are important in a lot of different ways. So they account for about a third of the ice-free terrestrial land area. They account for about a third of global NPP. They're a huge store of soil carbon, so they store more carbon than forests, about 20% carbon is stored in the soils. They're also really important in terms of humans and the intersection of human impacts on the environment. So grasslands are a really highly endangered biome worldwide. So about 70% of grasslands have been converted over to human-dominated land uses like cropping. Also, obviously, they're really important in terms of human food production. So a lot of the food we eat comes from grasses. A lot of the grazing land, these lands are used for grazing. They're also an area that has, sort of from a logistical perspective, they're actually really nice because the plants are small. So you can have a 5 by 5 meter plot and do a manipulation. If you're working on trees, you need much bigger plots, right? It gets more challenging. So there is a logistical part. Another interesting part about grasslands is, unsurprisingly, they're dominated by grasses, which if you've taken botany, grasses are all in the same family and they're very widespread. So it's kind of an amazing ecosystem where you can work everywhere from a salt marsh up to high elevation sites. You can work in deserts and tropical settings and still have one family dominating the whole ecosystem, and often a lot of the same genera. And so you can do some more phylogenetic and functional, you can basically parse that out. So a lot of other systems, as you move from system to system, if you're trying to compare shrublands to forests, it gets a lot more challenging. So we really like that from a scientific perspective. So this was our initial vision. So this really was like 6 or 7 of us. We were kind of starting new jobs. We thought we could, if we all did a nice simple experiment, then we could have this, which most studies are done at one site. So this would be like 7 times better, which would be really great. We didn't really picture this being a big global thing at all. And today we have sites all around the world. So every one of these sites is a research site, but you can look at these as sort of dots on a map. In reality, what this is, it's a global network of researchers. That's much more important than the experiment in a lot of ways. So these are hundreds of scientists around the world who've decided that they want to be involved in something bigger than their own work and put their work into a bigger context. They're all conducting exactly the same experiment, sharing their data, working on papers together. And so it's been just an amazing sort of social experiment in conjunction with an ecological experiment, I think. Don't you feel? You're going to meet great people, right? So there's over 170 sites now in 28 countries on 6 continents. There is a small grassland in Antarctica, but it's very complicated. We keep trying to get the 7th continent, but we haven't gotten there yet. Spans 130 degrees of latitude from almost all the way down at the southern tip of South America up to northern Norway. Every year we collect data, or the group collects data from about 4,500 5 by 5 meter plots. Currently we have about 350,000 individual species occurrence records that are available for people to work on. This includes data from almost 3,000 plant taxa from 150 families. I think this is like 1% or 2% of all the non-tree plant diversity in the world. It also spans really big gradients in environmental gradients. So in terms of human impacts, goes from basically no exotic species to 100% exotic species. Soils, very highly variable, so big gradients in soil nitrogen and soil phosphorus. Wide ranges of diversity, so some are almost monocultures, some have 30, 40 species per meter squared. Biomass production covers several orders of magnitude. Big geographic gradients, so we have sites all the way from salt marshes up to over 4,000 meters. And like I said, really wide latitudinal gradients, so almost 130 degrees of latitude, and big climate gradients, so from deserts all the way up to tropical systems, and temperatures from -10 mean annual temperature to 30 degrees. So we have that big global context. Sort of the way the way the experiment works is there's two sort of interacting experiments. So there's 10 treatments total. These experiments are in a completely randomized block design with 3 treatments, which isn't a lot of treatments or a lot of replication at a site, but a lot of our replication is really across the site. So this within site replication is more like what you would think about as a subsample, and our sites are sort of like your typical blocks, I guess. Your site is our replicate. So the first experiment is a multi-nutrient experiment. So we were interested in different kinds of nutrients and how they were affecting these grasslands. So it's a full factorial combination of nitrogen addition, phosphorus addition, and then a treatment that's sort of everything else, so potassium and micronutrients combined. So that's 8 treatments, all the combinations. Then we have a herbivore fencing treatment and an herbivore fencing treatment with nutrients. So there's sort of this multi-nutrient with 8 treatments, and then this top-down, bottom-up, which is a full factorial of with and without large herbivores and with and without nutrients. I was going to say something else there, but whatever. So we collect the same data at all the sites. So we have this core, so treatments. We also design it's easy to design expensive things, right? It takes a lot more creativity to design cheaper things. And partially we were being unencumbered by very much funding. We were, able to focus on some treatments that were cheaper, and we really focused on things that also scaled across the globe, not really specialized equipment. So this is really working with things that scale with the local economy. So basically it's just fertilizer and fencing, which most people can get pretty easily and sort of reflects the local economy. It makes it a lot easier for people to do. Also, these core variables are also pretty quick to collect. So we really, every time we wanted to add something more, we're like, we need to keep this simple so people can do it on the side when they're out doing their other work. So in every plot, so there's 30 plots at a typical site, 10 treatments times 3 replicates, collect the cover of each species, clip the above ground biomass, sort it into functional groups. We do light interception at ground level. And then we also aggregate together some site level things like site level climate, species level things like species phylogeny, the provenance of the species, so are they native or exotics, a little bit about their life history. The core, this has really become kind of a network, like that's pretty basic things to collect. We also have space in each plot where people can do additional studies, collect additional data. And this is really where most of our real work comes out. So we've published probably 150 papers at this point, and about over 80% of them involve, some sort of additional study that's done. These can take a lot of different forms. We have ecosystem things that are being measured, so soil texture, soil chemistry. We have studies looking at nitrogen and carbon flux, like Anita led a great one on N mineralization, plant litter decomposition, root biomass, also community responses. So we've looked at arthropod abundance, plant traits and stoichiometry, seed predation, soil seed bank composition, and soil and plant microbiomes. And these aren't done, all the sites do those core variables. These can be variable from 40 or 50 sites, or maybe even just 2 or 3 sites that one graduate student or postdoc does on their own. All right, so switching gears again, that's sort of the framework. So I'll go through and kind of give some key sort of highlights. Like I said, there's a lot of papers here, so this is really like going to be a little sampling of a few key points, but also sort of tracking them through time. So a little bit of the history across the last decade of some of the papers. So talk a little bit about what we've seen from ecosystem primary production, a little bit on consumers, so arthropods and soil and plant microbiomes, look at ecosystem diversity and stability and species invasions. And all of these are motivated out of both theoretical work and also a lot of the meta-analyses we're doing. So I won't go on and on about that part as much, but happy to talk about it later. But they're not just sort of random questions. They're questions for which there was either theory or individual site experiments that suggested these might be the case, but we didn't really know if they were general or how important they were. So things like, yeah, this happens here, but it's unknown whether it's a globally important thing. All right, so one of the first data sets that came out of this was we do pre-treatment sampling. So that actually, we hadn't really thought in advance that would be such a cool data set, but turns out just having 30 plots across the world that are in grasslands that are collected in the same way, there's a ton of interesting things there. One of the early questions we asked was just in those observational plots, what were the main drivers of above ground plant biomass or primary production? Of course, we all know from reading Ecology that the most important drivers are rainfall, right, and then soil temperature, but it turned out not to be the case. So this is a bunch of things that went in the model. So soil pH, soil N, PET, so potential evapotranspiration, rainfall, elevation. The biggest predictor is atmospheric nitrogen deposition. So this was a real surprise that like now in these grasslands, it's not, we should be like changing when we teach intro ecology, right? Like instead of MAP and MAT, it should be like, oh, it's nitrogen deposition. And again, here's just a graph of nitrogen deposition around the world. So already in these early papers, kind of a lot of surprises coming out. There's also a lot of theory that when you fertilize, you can lose species, and that's been shown in different sites around the world in individual experiments, but not in a standardized way. So you don't really know what's really happening. So here's a graph of species richness change in number of species per meter squared in the nutrient plot. So the fertilized plot, fencing, and nutrient by fencing, this dashed line is 0. So you can see when you fertilize, we're losing about 2 species per meter squared across all these sites. So that's an effect that's very standard across grasslands, marshes, montane meadows, pampas, whatever. Fencing had really variable effects, so not a net effect overall. So that gets at this idea of contingency. So some treatments have very consistent effects, some have contingent context-dependent effects. These species that are being lost, are these just a random set of species or not? We also looked at effects on species invasion. So here's a very similar graph. So this is showing richness change, but now splitting out the natives and exotics. So the natives are the open circles, and the closed circles are the exotics. So when we fertilize, we're only losing native species. We're not doing anything to the exotic species, but this is cover change. We are increasing the cover of exotics. So it's not just a random species loss. We're causing increased invasion and loss of native species. And this was actually really surprising in a lot of ways because there's hundreds and hundreds of invasive or exotic species here. So there is some consistency, even though these are hundreds of species from dozens of different families, but there is a selection where they're doing better under high nutrient regimes. And the same with the natives. There's thousands and thousands of natives. The natives are very sensitive, even though they're from all over the world. So getting into that contingency effect, so we didn't find an overall effect of fencing on species loss, but that doesn't mean it's not important, it just means it's variable. So here's a graph that shows the effect of fences on richness. So 0, every one of these dots is a site, so individual sites all around the world. So in some cases, the fences cause, when you put up a fence and exclude herbivore, you lose species. In some cases, you gain species. But this is really related to the effect of the fences on light. So over here, when you put up a fence, you get more shading, you get a lot of biomass being built up, and those are the cases where you lose species. If the herbivores are, if you put up a fence and there's no effect on light, then you don't have much of an effect on the, and when you think about this more and look at it more, you can really find that these herbivores can sort of overcome some of the eutrophication effects. So these grazers are reducing that negative effect of fertilizing, which is driven by increased shading. So this is looking at this top-down, bottom-up experiment with the nutrients by herbivores. We also have that multi-nutrient experiment, right, with the nitrogen, phosphorus, potassium, and all the other stuff. There's a lot of interesting theory that suggests not just when you add nutrients, but when you add multiple kinds of nutrients that you can lose more species. So a lot of organisms generally, but plants have trade-offs in the nutrient use efficiency. So they might be more efficient in using nitrogen, phosphorus, water, on and on. So as you add more kinds of nutrients, you collapse down the opportunity for those trade-offs. And we found that to also be the case. So here, this again is diversity change. So this is 0. This is the number of added resources. So 0 is control. 1 added resource is nitrogen alone, phosphorus alone, that kind of thing. 2 is like adding nitrogen and phosphorus together. 3 is adding all of the nutrients at the same time. And you can see as you add more and more kinds of nutrients, you lose more and more species. So that maps really nicely onto some really simple ecological theory, and was a really great demonstration of this that had been shown at 1 or 2 sites. Surprisingly worked really well for basic theory, which seems to often not be the case. That diversity loss in some experiments has also been linked to changes in stability. So sometimes more diverse experiments, especially if you set up a diversity experiment. So if you plant different numbers of species, a lot of times those species can be asynchronous with each other. So some species are high in some years, low in other years. And as you have more and more, you can have this sort of averaging effect where every year you can have a more consistent production. That turned out to be the case in this experiment as well. So this is the average number of species in the plots. So going up to 40 species. Every different colored line is a different site, and then the black line is the mean response. So this is the stability of productivity, stability of biomass. So this is basically how variable is the biomass if you measure it across multiple years, between years. And you can see overall and at all the sites, as you get more and more diversity, the sites get more and more stable through time. This is looking at a measure of what's called asynchrony. So if the species are all moving up and down together, then they're highly synchronous. Asynchrony is a measure of how out of phase they are with each other. And so you can see as you have more and more species, they become more and more asynchronous. So if you just have a few species, then they also move together. This is kind of observational data, but then we could look at the nutrient effects and really big changes. So when you fertilize these plots, you not only lose species, but you lose this coupling between diversity and stability. So here's that dotted line, is this line from here superimposed, but now there's no longer any relationship between the diversity of the plots and the stability. And the reason this is occurring is because we've broken down that asynchrony effect. So now, no matter how many species you have, you've selected for species that move in conjunction with each other. So we no longer have this asynchrony, which leads to stability. All right, those are all about plants, which are very interesting, but there's other organisms in the world that eat plants that we need to pay attention to. One add-on that we did early on was doing arthropod sampling. So we had people at a number of different sites out with, you know, those leaf blowers that are really annoying. You can turn the fan around and turn them into leaf suckers. So we vacuumed up all the insects, and then we weighed them to get secondary production. So here's a plot showing arthropod biomass in all of the treatments. And all the treatments, the high ones here are nitrogen, nitrogen and potassium, nitrogen and phosphorus, and nitrogen and everything else. So all the nitrogen addition treatments have higher insect biomass. And this is the fencing experiment that when we fertilize or fence and fertilize together, we get more insects or more arthropods. That could be a number of different pathways. So we worked on pulling some of the data to produce an SEM. So here we've got our different treatments: nitrogen, potassium and micronutrients, phosphorus, and fencing. All of these can affect plant biomass, the structural diversity, can affect plant quality, and by that I mean the nitrogen content. So there's a mismatch in stoichiometry generally between animals and plants, where animals have a higher nitrogen content. So as plant nitrogen goes up, you have less of a stoichiometric mismatch and plant diversity. And what we found, so nitrogen increases plant biomass, increases the nitrogen content of the plants, and reduces diversity. But the really strong links are between this nitrogen measurement, so plant quality to the arthropods, and arthropod body size. So we're not necessarily getting more insects, we're getting much bigger insects, and that together produces the production. So basically when we fertilize, we're moving from lots of little tiny things to the big giant grasshoppers and things like that. So provided a lot of, you know, interesting insight about how stoichiometric change is actually one of the big links that's happening here, not just producing more plants. We also have been doing a lot of work on microbial communities. So this was our first look at soil microbiomes. So this was another really nice add-on where we had people at a whole bunch of sites. They collected soils, overnighted them to one lab. That lab pulled out some and froze it for DNA extraction. Also looked at root biomass, soil nutrient pools. What else did we do? PLFAs, a lot of different measurements in the same soil core. And what we found across all these sites is that these fertilization treatments are having really consistent effects on big groups of the soil microbiome. So the columns here are fungi, archaea, and bacteria. This is the effect of nitrogen and phosphorus. So the blacks are the controls. So these are just ordination plots showing the community change. So you can see for fungi, archaea, and bacteria, for nitrogen, the blue is very easily seen as being separate from the black, and then the same with the phosphorus. So we're getting these consistent changes in communities all across the world on this soil microbiome. Since we have the plant community at the same time, we could also compare the effects of these fertilization effects on the plant community composition, and found that the plant community change is tightly coupled with the soil microbiome change. So here is the plant community compositional change, and then the microbial community change for bacteria, fungi, and archaea across the different, so any of the treatments, and then the colors are the different continents. And for both bacteria and fungi, we found that as you get bigger and bigger changes in the plant community, you also get bigger and bigger changes in the microbial communities. Using those same data, so that's microbes, there's like a gazillion microbes. Do they actually matter? We can pull some of these microbes together into broad functional groups. So this was a follow-up paper a few years later looking at the same set of data, but now taking two parts. One is taking the soil microbial community, fungal community, and breaking it into mutualists like arbuscular mycorrhizal fungi, and then in the soil, the mycorrhizal fungi, pathogens in the soil, and saprotrophs. And we also took our root samples and did microscopy, so we could look at both the colonization in the roots, especially for the AMFs, and then also in the soil. Here's a treatment to control nitrogen, phosphorus, and nitrogen and phosphorus together. So what you find are that when you fertilize, when you add nitrogen and phosphorus together, you lose these important mutualists in the soil, so the arbuscular mycorrhizal fungi. And when you add any nutrients, so nitrogen, phosphorus, or add them together, you get increases in soil fungal pathogens. Also did a project with leaf microbiomes, which was also really interesting. So that was another one of these add-ons that we mailed from our lab, a whole bunch of falcon tubes to people all around the world with buffer in them. Everyone went out with scissors and put the leaves into the falcon tube, seal it up, mailed it back to us, and then we did the sequencing in our lab and have been finding really interesting results. Kind of a first cut through this was looking at effects on diversity, and what we find for both fungi and prokaryotes is that when we add nutrients, we get increased diversity in the leaf microbiome, and when we fence, we also get more fungal diversity. And when you look at this more deeply, what you see the big mediator is increased shading. So when you fertilize or you put up fences, you reduce light, you get these more shaded sort of microclimates, and you're getting more diverse microbiome communities. We're just working on a compositional paper on that now. Luis has seen the early, it's going to be cool, isn't it? Yeah. All right. So switching gears, and this is sort of a prequel to Tuesday, to hopefully you won't be sick of listening to me talk a lot. But so this is thinking about spatially extensive work. The other big dimension that we don't do very well at generally is looking at long-term experiments, because they're also very hard to fund for a similar and different set of reasons somehow. So in the same way that thinking about if you do an experiment in one place, does it tell you what happens around the rest of the world? You can puzzle over whether if you do a short-term experiment, does it tell you what happens in the long term? And this is really, in a lot of ways, related to funding cycles. So in a lot of places, you get a grant, it's for 2 or 3 years, or you have you're a student trying to work on your project. You're not going to do a project for 30 years, or your advisor is going to be like really annoyed with you because they're going to retire before you finish. So you can think about this. So here's treatment years. I just drew a line at 3, which seems like kind of the standard NSF grant for us. So with the gray line, if you have this case where this is just a log treatment over control, you have some response where you have a response in 3 years and then it just stays flat. That's, then you can make that prediction and say, well, we did a 3-year experiment, then we're good. We understand the system. But that's almost never the case. And some scenarios that you can have arising, you can have these kind of slow systems where change kind of builds up over time. So there's not much, and it just gets stronger and stronger. You can have a lot of cases with buffered systems. So you might have a case where you can perturb it and perturb it. You can do this for 10 or 20 years and nothing will happen, and all of a sudden you hit some threshold and the whole thing starts to switch over. You can have negative feedbacks. You can easily imagine, like you do something and it's like, whoa, but then it just fades away. And you can have these really variable systems. So obviously climate in particular is going up and down. You can have oscillations in herbivore communities, or pathogen outbreaks, and that can also make it really hard to pull out a signal. Like you might find nothing, but really it's just because you need more time. I'm going to talk more about this and give some examples from our Cedar Creek site where we have experiments that have been running for 20, 30, 40, even 60 years, and show demonstrations of all of these kind of cases. But I don't think I'm ever going to show this case because I don't know that we've ever found that one. We're still waiting for the thing that behaves itself. But I want to just give one kind of example because now this nutrient network, we've all been working on it long enough where we now have sites that have collected, like our longest running sites now are collecting their 17, 18-year data. We have sites that have 12 years of data. Like we're really pushing this limit. So now we can start asking, these long-term experiments are very rare. Instead of having one or two of them, now we can say, what are the cases that cause us to have more buffered systems or have us these strong negative feedbacks? Do these things always behave the same way over time, or are there sites that are slower, faster, more resistant? Kind of a first look at this was looking at the years of nutrient treatment. So this is just all nutrients or control. This is the 0 line. And this is going back to some of the stuff I've showed you. So this is the effect of nutrients on above ground biomass. So the black line here is the average effect, and all the other colored lines are all these different sites around the world. And you can see this is kind of that slow system kind of thing, right? Where as you add nutrients for longer and longer, out to 10 years, the effect just keeps getting bigger and bigger. We had exactly the same thing at almost all sites for effects on plant diversity. So you can see that basically all these sites, the effects on plant diversity are dropping, though some kind of saturation going on, and a little variable. Like we have some crazy kind of sites. I think that's Kansa. But anyways, this is an area that we're just kind of pushing the frontier here to sort of merge together really spatially extensive with really, really long temporal data and understand what are the drivers of some of these long-term trajectories. All right, in summary here, okay, I'm doing okay. So just kind of some scientific parts. I think that there's a, hopefully I've made the case that there's a need and also a way to confront some of these canonized concepts. So there's some famous experiment, and you're like, oh, this must happen, but we should really be testing this using experimental data. The same with theory, right? We can have theory, we can test it at one place, but that doesn't mean it's globally relevant. Some of the key things we found that eutrophication will lead to loss of native diversity and increased species invasions, and also a lot of other microbial effects, increased pathogen loading in the soils. Herbivores can counteract this loss of diversity when they act on increasing light penetration, and that eutrophication weakens the stabilizing effects of diversity. That's kind of lots of, just a snippet of those. More on the methodological side, I guess. These distributed experiments can be a real complement to traditional approaches, so I'm not making the case that this is by any means the only way to do. I think all of us need to do multiple kinds of approaches because they all have trade-offs. So whether you're doing modeling and observational studies and experiments, meta-analyses, all these are really great, and they all can provide insight, and they all have annoying parts about them. But it's an important complement. Also that these big collaborative studies are possible without massive budgets. So it can feel really disempowering. You have these, like periodically, there's these huge studies that show up. Like this also came out of a time when in the US there was this huge investment in this network called NEON, which got just hundreds and hundreds of millions of dollars, and as like regular people were like, well, we can't do, we don't have that money. We're never gonna have that. But using some creativity, you can actually do some really compelling work. And there's been a lot of really clever studies that people have done that have done this. Focusing on sort of general treatment. So like I said, this isn't something where you can really get into the details of your study site and really finely tune things. And actually at a lot of study sites, people will layer on other treatments. Like at Las Chilcas, there's an extra mowing treatment and a cattle grazing treatment that are added on. At our Cedar Creek site, we added a bunch of nitrogen treatments to get a nitrogen profile because it was something we were particularly interested in. And if you're interested in joining NutNet, talk to Laura and Pedro. Or start your own network, even better. If you are going to start your own network, you want to make sure, and you can look at this paper that Elizabeth Borer led, kind of a like do-it-yourself manual for starting your own network with all of your friends. You want to make sure you have clear scientific goals and questions. This having identical treatments and sampling approaches is really important, and something we sort of spent a lot of time catching up on because it was so novel is just really having clear ground rules. People need to know and have confidence and trust that they're putting a lot of their effort in, and you are, you're paying a little bit of a price, and that you're giving up some of your autonomy. It's like, okay, I'm going to wait. I'm not going to rush my own paper out. I'm going to wait for these network things to arise. You need to set up a trusting network in a way that people feel confident that, they're doing this work year after year, and it's going to pay off at some point. But it's a little bit different than just blasting through with your own work. Think about simple and inexpensive designs. So it's really easy to sit down and come up with a $100 million project. A little bit of creativity, 2-liter bottles of Coca-Cola, and you can do lots of interesting things, right? Leaving room for additional studies is really important. We have a paper that's going to be coming out in Frontiers in Ecology and Evolution, looking at a lot of these add-on studies I talked about. It should be out really soon, and some sort of guidance on how to do that without messing up the other experiments or annoying your collaborators. Needs to be sure that there's really clear benefits for participating scientists. So nothing frustrates people more where they join on, they put a lot of their limited resources and time into something, and then nothing happens. Like, that's really not a great experience. And first and last is data management. So having really, we got a little bit behind the game here and had to play catch up, but having really clear data management, we have a couple papers on approaches for this, is really important at the outset. I would be remiss if I didn't mention this new network, because if you have one global network, why not have another one? I think 2 is enough. This is a network which is looking at disturbance and recovery across grasslands. So this is taking, everything's the same in terms of the sampling and plot sizes, and it shares the control and fertilization treatment. Whoa. Okay. And but now it's also combined with a disturbance treatment. So we do rototilling for 3 years in a row and then stop. And also we're looking at recovery from fertilization. So we fertilize for 5 years and then stop and look at recovery. So this has been really exciting. We have over 60 sites in, if I had my notes, some number, 15 countries, I'm gonna make it up. The really exciting part, this, and also DroughtNet, which has hundreds of sites. We've all been coordinating across these networks. They're using the same sampling. We've all shared the same database, so we're all using the same database. So all of these now are interoperable. So once these networks are kind of doing their own work, we're setting the conditions where we're going to have a meta distributed experiment, which is going to be really exciting. Like 250 sites with drought and disturbance and nutrients and grazers all across the world. Wow. It's going to be great. So acknowledgements. It's funded by Fulbright, University of Minnesota, the Cedar Creek LTR, Long-Term Ecological Research Site, which I'll talk about on Tuesday. National Science Foundation has been critical for this. Lots of coordinators, so NutNet coordinators. We have postdocs, Eric Lynn, lots of other people have helped us. Lots of collaborators working together, especially coordinating committee, people reading all these abstracts that come through, like Pedro, and all the other NutNet researchers. This is really a collaborative project. It's about hundreds of people working together. And yeah. All right. Thank you. Happy for any questions.
Yeah. Bueno, no tenemos demasiado tiempo, pero pueden hacerlas en español y hacemos el intento de traducirlas. Si es que no se animan a hacerlas en inglés, no hay ningún problema. Pero bueno, también después del seminario nos quedamos a comer en el comedor con Eric y con Anita y Martín, así que también están invitados a eso. Estoy haciendo tiempo para que levanten las manos. Preguntas para Eric. Sí.
In the first slide, you showed a graphic about the importance of the different influences of human in the ecosystem.
Yeah.
And I wonder how you measure that. It was the
Oh, I didn't write that paper. So there's been a couple papers. So that was by, I forgot his first name, but Stefan, and there was another paper. Rockstrom, thank you, yeah. So basically they tried, instead of just, there's a lot of, what do humans do, but they tried to kind of get this idea of a planetary boundary. Like you can perturb systems, but if you go too far, then they can't recover. So they just collected lots of data in the literature, and, you read it, like it's hard to do, but kind of the point is there are things that we know are like way out. There's a lot of things we don't know, but yeah, it's a cool paper. I thought it was a cool idea. But
thank you very much.
yeah.
Sí. Felipe. Perfecto.
Veo que trabajan en los Grassland, que es un ecosistema muy importante y que está distribuido en todo el mundo, y creo que tiene muchas ventajas ese tipo de ecosistemas. Pero los bosques también son sistemas importantes y muy castigados, muy transformados. No criticando esto, sino preguntando cómo podríamos hacer para, o si ya hay redes vinculadas con ecosistemas boscosos, sobre todo nativos.
We want to receive.
So,
You help me?
yes, he wants to know if you have in mind some network related with forest, similar
Yeah.
network. So también experimentos.
Yeah. There are things in other ecosystems. So NutNet had projects in forests. So that one did. We go into sort of savanna systems with kind of nutrient network, but a lot of it's just about the scale of the individual plants. Like our plot sizes are small. There are global tree diversity experiments. So what is it called? IDENT. Has those aren't really standardized, so they're more kind of semi-standardized but integrated together. And then there's also what is it called? Forest Geo, right? These like 50-hectare observational plots which are standardized. I can't remember. There's some big standard size, and you go out and with your tape measure, and you measure all the trees every 5 years. I have friends who do it, but. So there are projects, but for us, like the plot, we like the plot size.
Sí. Última.
I missed your workshop, so I'm asking a question about that.
Yeah.
And in the list of conditions to set up a network, I think you skipped an ion critical mass.
Yeah.
What's that?
So, we had a group of people that we knew already. So there was 5 or 6 of us, and we were like, if all of us do this, then it's going to be great. Like we didn't have to wait to have it be successful, to have, okay, let's wait until we get 50 people. So that's kind of what I meant. Like if you talk to a number of different collaborators, even 3 or 4 people, and they all agree to do the same thing, then you're like, okay, if we all do this, there's going to be a cool paper and it's going to be exciting.
An initial commitment.
Right. And so just kind of locking in one, so you can just go ahead and say, okay, this is enough to have it be really good, and then if it's more, that's better.
Bueno, le agradecemos